Okay, sorry about the delay there, I was just trying to set that up. Okay, so in the last lecture we went through the Michaelis-Menten equation. We went through how the logic of it, how it comes together. The idea that we're just measuring the, it's a way of figuring out the velocity from a reaction. We went through how we work that out, how we can measure the product made over time, which is velocity, and how the product made over time increased as we increase the concentration of the substrate, obeying first order reaction kinetics, which was just normal velocity equals rate multiplied by substrate concentration. Velocity equals rate, it's a lowercase k, multiplied by substrate concentration, normal first order reaction kinetics. And really how this all links up together, that you can keep adding substrate and it will keep increasing the velocity up to the point where you hit the maximal velocity where it can't increase anymore. And also defined as well Km. Km being the concentration of substrate required to hit the half maximal velocity. And can anyone tell me why was Km important? What is the value of Km? What can we, if we're talking to each other as biochemists, what is the value and importance in talking about Km values?
If it is
That's
low,
the, yeah, but, sorry?
if it is Km is low, so that enzyme have high affinity for substrate.
Yeah, so it's communicating substrate affinity, substrate preference is often the one that's used, whereby a lower Km value is suggesting a stronger affinity, because if it's the concentration required to hit half maximal velocity, if less is required to hit half maximal velocity, it's kind of like saying it's a more efficient fuel in a roundabout way. The reason why we do it at half maximal instead of maximal is because we want to catch it in the logarithmic phase. So it's just a way of, it could be the quarter maximal or the 3/4 maximal. They just set it to half maximal so you can catch it in the logarithmic phase rather than the max phase, because sometimes the max lags when it transitions from the log to the steady. It might lag a little bit, so we want to catch it half maximal where it's still logarithmic. That's all that it is between it. And yeah, how they're all linked together. Now we touched on a few little bits that I kind of finished, but I'd like to just remind you of. There's some algebra that kind of explains the reason why Km is equal to half maximal the concentration required to hit half maximal velocity, and that's just written in there, but that's not super, I'm not going to go over the algebra, just in case you were interested. And to mention as well, this table, which is, this is a table that is just for information, it's a table for flavor, it's not a table to memorize. For instance, if we were to look at hexokinase, and it has multiple substrates, what is the between, ATP is always a substrate for hexokinase because it needs the ATP for the phosphate donation. But between glucose and fructose, what is but going by the numbers there, what is the preferred substrate? Glucose. Why? Lower Km. So it actually will react with fructose, just a lot slower. What's that, 30 times slower? So it's much more effective to work with glucose. But just to look at what kind of numbers we're looking at between. And broadly, it's just a measure of substrate affinity. Also talks a little bit about Kcat. That's the number of molecules that are converted into product per second. And it can be very, very fast. In fact, it gets to the point where an enzyme can work so fast that it's at the theoretical maximum speed that a chemical reaction can occur, and it's only limited, not the theoretical maximum that a chemical reaction can occur, that's nonsense, but I'm saying that it is limited by, not by how fast the reaction can occur, but how fast the material can diffuse through space and time, or just space. The idea that the reaction occurs so fast that it takes longer for the stuff to dock with the protein and then leave the protein than it does for the actual chemical reaction to occur. And extremely efficient. You're only limited by the physics of how fast matter can move because the chemical reaction is so fast. When you get up to things like catalase, which is extremely rapid reaction. And this is how it is calculated. Written in the red is how it's calculated, because this here, is just this. Velocity, but just don't worry about it being the Vmax and the E total for a moment. but just velocity is equal to the rate multiplied by the concentration of the substrate. Well, if we just say that in the reaction of E plus S makes ES, and then ES to EP, and then EP to break apart again. If we're interested in that middle bit, which is how fast the reaction works, that's ES to EP. That's the middle bit. I'll write it down. If we're interested in this E plus S to make ES, to make EP, to make E plus P. If this is the chemical reaction taking place, then we want to look at the rate of going from ES to EP, which means that kind of what we're looking at is the velocity is equal to the rate multiplied by the concentration of ES. Because in this point, this is reaction 1, reaction 2, reaction 3, and in a roundabout way, the substrate for reaction 2 is ES. So we can just substitute in ES. Joel?
So is it called rate limiting step reaction?
So usually, yes. Yeah. The reaction 2 is usually the rate limiting step, which is why we're interested in measuring it and why we would measure it in the pre-steady state. And so if we want to measure what is going to be the K value, an easy way of doing it is that when you have ES, saturated, what kind of rate value have we reached? What rate value have we reached when ES is saturated? Vmax. Right. Vmax. And when ES is maximal, it is equal to E total. That's it. You got it, guys. So Vmax is equal to the rate multiplied by E total. How will we rearrange this? Simply just to make the K value, the rate, the subject. K equals Vmax divided by E total. That's it. And so if these chemical reactions that we're doing with the enzymes, like when I drew out that graph at the beginning, and remember that you chuck in a bunch of enzyme, and you'll get to a point where it just becomes, you keep adding substrate, and then the reaction will no longer speed up because it's becomes saturated. Well, how could you then make it speed up more? Well, you could put in more enzyme, because the enzyme that's there is saturated. But if you put in more enzyme, then you've got more enzyme. So you've got a new level that it can get saturated up to. So if you do Vmax divided by E total, that'll be the maximal speed of the reaction divided by how much enzyme you have to find the rate per enzyme in a roundabout way. And therefore, the units are going to be in per second. So it'll be the number of molecules per second which are converted. So to do this is there a homework on it? There's a homework on it, right? There's a homework on being able to do this out at the moment, and then as well as that, it'll be a question you'll be expected to do in the exam. So using this equation and using that in the red are two things that are going to come up they're in the homework and they're going to come up in the exam as well.
Wait, what was the first one?
Just
That one.
that.
Are you expected to know the equation or to actually like apply it for the exam?
Definitely have to apply it. I don't know, I mean, for grad students, I probably expect you to memorize it because it's just V0 equals two things divided by two things. It's not, I don't know, like it's not a math class, so if it was super difficult algebra, then I'd probably give you the equation. But if it's just two things divided by two things, I'd probably just expect you to remember it. I think that's, if it was much more complicated than that, I might give it to you, but I think that's simple enough that I want you to memorize it. Okay. And then just one final thing that I didn't get to, was to just say that if we have the KCAT, that'll tell us the efficiency, the maximal efficiency that an enzyme can have. And then the KM tells you the substrate preference. So that allows you to compare different substrates within the same enzyme, but different substrates within the same enzyme. But what if we wanted to compare different enzymes and different substrates? You can do the KCAT value divided by the KM value, and that allows you to compare enzymes to find out which is the most catalytically effective and rapid enzyme for this value here. And this comes up where it can get really close to like the upper limit, like I said, the upper limit of how fast a chemical reaction can actually occur, because you're no longer limited by the chemical reaction, but actually just the speed of matter coming together. When it gets up to these numbers up here, they're just like super fast. Like acetylcholine esterase is such an effective enzyme that it has such a strong affinity for its substrate and breaks things down so rapidly that it's just considered to be a near perfect enzyme. And so perhaps you could use genetic engineering to make an enzyme more perfect, where it has a stronger affinity for its substrate, Km, and a stronger, more rapid speed rate at which it works, which is the KCAT value. And I think that's really where we get up to with the kinetics. We're now going to shift gears actually and go back into mechanics. We're going to go into mechanics in a little bit more detail, and actually then that will conclude enzymes. So we're almost through the woods of the worst of it. Okay. So just to mention, when it comes with mechanics, we've done simplified things. We've just done one substrate comes together with the enzyme and forms one product. That doesn't happen all that often. Actually, what does happen very often is that about 2/3 of enzymes actually have two substrates and two products, or multi-substrate. I just have to mention that really how that happens. For example, you can have a group is transferred one substrate to the other. So substrate 1 comes in, leaves something behind, and then substrate 2 comes in and picks it up and takes it. Like a group transfer. Like maybe, like for instance, with a phosphorylation. ATP comes in, drops off a phosphate, and then leaves as ADP. And then something else comes in, picks up that phosphate, and leaves as the phosphorylated form, where it just takes like an order. That's a very common type of reaction. Or you can have a different type of reaction where two things come in, one gets oxidized, one gets reduced, and then they leave as the respective oxidized or reduced version of each other. So for instance, NAD comes in and leaves as NADH, while at the same time something else comes, there's tons of different things, something else comes in, let's say pyruvate comes in and leaves as acetyl-CoA. So one thing gets oxidized, one thing gets reduced because there's a transfer of electrons between them. So just to briefly mention that, of course, there's a few different ways that can happen. It can happen, this is an example which I like, but these two are really the key two ways in which it happens. So the first one is you start with the enzyme. One substrate binds, a second substrate binds, and then those two, so they've got two substrates bound at the same time. Those two substrates are turned into products, and then one leaves, and then the second one leaves. An example of that, well, it's actually, it's kind of like that, would be, for instance, step 1 of the TCA cycle. Does anyone remind me, what's step 1 of the TCA cycle? Yes, that's part of it. Sorry?
Oxaloacetate and acetyl-CoA.
Yeah, citric acid synthase. Citric acid synthase, oxaloacetate and acetyl-CoA come together to form citric acid. So in that case, what you have, you start with the enzyme, citric acid synthase, and then first of all, oxaloacetate binds. It's always oxaloacetate binding first. Oxaloacetate binds first, then acetyl-CoA binds second, and then they are condensed by an aldol condensation into citric acid, and then that citric acid leaves. Actually, the citric acid leaves and the CoA group leaves as well. So you get that two things go in and then two things go out, but they go in at the same time. So that's the ternary complex. That's the ternary complex that's in A example. A different example would be where there is no ternary complex, so you don't have substrate 1 and substrate 2 binding at the same time. Rather, substrate 1 comes along and binds. It then turns into product, but when it turns into product, it leaves something behind. Like the example I gave where ATP binds as the substrate, it turns into ADP and leaves behind a phosphate, and it's attached to the enzyme. And that's what that little apostrophe there means. It means the enzyme has been modified because it's now inside the active site, something is phosphorylated. That product, which is ADP, then goes and leaves. The next substrate comes in, which is the thing to be phosphorylated, and takes the phosphate group. When it takes the phosphate group, it restores the enzyme to being what it was, and it leaves as a phosphorylated product. So that's the other example. There's many other different ways in which it can occur. Those are the two most common and simplest ways in which a multi-substrate enzyme functions. Edie?
First A, does the binding of one substrate affect the binding of the...
Very much so. Very, very much so. The question was, does the binding of one substrate affect the binding of another? Yes, absolutely, very much so. Such as the example I gave with citric acid synthase. Oxaloacetate has to bind first. Because oxaloacetate binding will change the shape of the enzyme so that acetyl-CoA can bind. For two reasons. The first one is that when it catalyzes the reaction, if you think of the active site pocket, it always goes as oxaloacetate and then acetyl-CoA, and it has to be in that order, because if it was the other way around, then it would be like that, and that wouldn't be citric acid. It wouldn't make the same molecule. It would make an isomer of citric acid. If this is the acetyl-CoA, it has to go in the correct order, otherwise it would make a different molecule. The other thing is that oxaloacetate is often the limiting factor, and so you don't want acetyl-CoA bound, sitting around, waiting around, because there isn't any oxaloacetate. So you want to say that if there is sufficient oxaloacetate, then only then will the acetyl-CoA bind. If there isn't enough oxaloacetate, saying, hey, there's not enough of this, let's just not, and do something else instead. So it's a way of only using your substrates when you need and want to use them. Another level of control that you have as an advantage of enzymes that other things don't have. So good question. And this is an example here. This is literally copied and pasted from my PhD thesis. I want to find a way of wedging that in. And this is an enzyme. This is a mechanism called ping pong bye-bye. And it's literally called ping pong bye-bye. I've not made that up. And the idea is that along the bottom is the enzyme. And you see that you start with the enzyme in the PLP form, and you end with the enzyme in the PLP form. PLP is vitamin B6, pyridoxal L-phosphate. You start with the enzyme with its cofactor, PLP. A substrate comes in, ping. It modifies PLP into PMP, and then leaves as a keto acid, pong. That's the ping and the pong. And you see this amine group here? This amine group is oxidized to a carbonyl group. And it's a ketone, and this is the keto acid. So an amino acid is broken down into a keto acid, and it leaves behind that amine group. The M is for monoamine. And then it exists, the enzyme's in the monoamine form. Then comes along a substrate keto acid, ping, picks up that PMP. I reckon that probably would have been better written as PLP now that I'm looking at it now. But I guess I'm saying here that it's a complex. I guess it does, because it's a complex and it is all one for a moment. And then it leaves as the amino acid, pong, and restores the enzyme back to the PLP form. So it's and it's called bye-bye because it's 1, 2, bye is 2, Ping pong, bye bye. That's what they call it. But that's just an example of one. Okay, now we're going a little bit back into kinetics, just to make it easier to talk about inhibitors. So this plot from earlier, this one here, that plot has a curve. And now with modern technology, we can plot curves and we can do non-linear regression. And we could make, if we've just got several points on this curve, we can make a guess at where the other points are using non-linear regression. You may think that it's quite easy. You may think, well, hey, I can just draw in some points and just draw them in yourself. But that's you using your brain power and making guess at where the points are in between. Now, if it's a straight line, you do point A to point B, and you draw a line between them, and everywhere in between is going to be 100% accurate because it's a straight line. When it's a curve, all the points in between, you're just making guesses. And so modern technology can make guesses. If you've just got 1, 2, 3 point, modern technology can make the guess pretty good. But it's actually fairly recent. And years ago, what we would do instead is that we would take that curve and turn it into a straight line. And then straight lines are always perfect. And to turn a curve into a straight line, you can do a mathematical trick where if you just do the reciprocal of both sides, and that'll be a double reciprocal, it generates a straight line. And it's just, if this is V0 and this is S, if we just do 1 over V0 and 1 over S, it turns a curve into a straight line. And that's what a Lineweaver-Burke plot is. If you notice, on the y-axis is V0, on the x-axis is the substrate concentration, and we can derive Vmax and we can derive Km. If I go forward to the Lineweaver-Burke plot, on the y-axis we have 1 over V0, on the x-axis we have 1 over S. So the axes are the same, measuring the same thing. Different scale, but measuring the same thing. We can also, from this graph, derive Vmax and derive Km. And we just do 1 over Vmax, and that will give us Vmax. Or the slope of this is equal to Km divided by Vmax. So the idea is that if we only have some of the points of the data, if we just have this point, and this point, we can just draw a straight line between them and then figure out the rest of the, know the rest of the points because it's a straight line. But also, that now it's a straight line, we can use the straight line equation, y equals mx plus b. So we can figure out any point on this just using simple algebra. It won't be perfectly accurate, it will be slightly off because we're doing a derivatization, but it'll be fairly accurate. And until modern modeling techniques was the most accurate way of doing things. When I say modern, I mean like 30 years ago, but still, you know. But it also is still kind of useful because to teach now, because it can, the shape of the curve, or the shape of the lines can give us information that is actually very quick and easy to figure out and can tell us information about inhibitors. So I'm just telling you about the double reciprocal plot just to set up, when you compare, this is just a normal reaction, and when you compare it to an inhibitive reaction, it'll give a special shape. And from that shape you can tell the type of inhibition that's going on. That would normally be fairly difficult to do, so it's still useful today. But like I say, you essentially just, if you were to do this experimentally, what you would do is that you would set up a reaction where you have your enzyme, you have a set concentration of your enzyme, and then you would add in some of your substrate, and then watch a known concentration of your substrate, and then you watch to see how much product it makes over time, and then look at the initial velocity. Then you would repeat that experiment, but do it with a higher concentration of substrate. Then repeat the experiment and do it with a higher concentration of substrate. And every time you do that, you're making a different point on this graph. You start at a low concentration with a low speed. You add more and it'll go faster. You add more and it goes faster. Because it's a reciprocal, it's the other way around. So this is more and this is faster. So this is getting faster as you add more, going down like this. And so anyway, so you'll notice that as you add more, it goes faster. And you could plot it on a curve, or you could find the 1 over each number, 1 over whatever the substrate is, 1 over whatever that velocity happens to be, being the product made over time, just like speed is the distance over time, and then plot it. And that will be really important for inhibitors in a moment, I think in 2 or 3 slides. Just to set up that a little bit, just to make sure that you guys know that the structure of the enzyme is important for its activity, as we kind of change gears into enzyme like regulation of activity. That if the pH, for instance, changes, then the enzyme will lose or gain activity. For instance, pepsin. Pepsin is an enzyme that's in your stomach that helps break down food, and so it functions. If this is function and this is pH, it functions best at a low activity. And as we increase the pH to get to a neutral, close to a neutral pH, it actually loses its activity. So it works best at a low pH. Whereas glucose 6-phosphatase, which is an enzyme in gluconeogenesis that's in your liver that turns G6P into G to go out into your blood so your other organs can take it up. That works best at a neutral pH because the cytosol is a neutral pH. So enzymes will be adapted to work best at the pH that they're supposed to work at for that environment. And it may just be that this has got certain amino acids that are maybe carboxylic acids that at that pH are going to be protonated. But at a neutral pH will be deprotonated, and so that's going to change the bonds that will form. They'll change the shape of the enzyme, they'll change the activity of the enzyme. Again, to say that enzymes can be regulated, and this is important because you don't always want your enzyme to be on. Sometimes you want it to be slower, sometimes you want it to be off. That's all there is to it. There's a few different ways of regulating enzymes. One of them is with inhibitors that we're going to talk about in a moment. The other one is it can be regulated allosterically, like we did before with hemoglobin. Hemoglobin is allosterically regulated because nothing competes for where oxygen binds. Actually, no, sorry. Carbon monoxide competes for where oxygen binds, but the rest of the things like BPG, CO2, and acid, they don't compete for the oxygen binding site. So those are allosteric regulators. Carbon monoxide would be a type of inhibitor actually instead. But those are mostly allosteric. There's also as well different things like, for instance, reversible covalent modification. That's, for instance, where a protein gets phosphorylated and that switches it off. Or there'll be binding of separate regulatory proteins. That's where a separate protein comes along, binds to it, and stops the first protein from working properly. Something like, for instance, P53. P53 binds to other proteins and makes them work faster or slower, switches them on and off. So that's another thing, things can bind to it. And finally, this last one, removal of peptide sequences by proteolytic cleavage. I don't think I talk about that one in detail in this class, but there are some proteins, which is kind of interesting, where they have the protein, the full length of the protein is inactive. The protein becomes active when you chop a little bit off the end. So it's actually the full version doesn't work, but the chopped version does work. And those are activated by proteolytic cleavage. Proteolytic means proteo, proteinlytic means the cleave. Those are things called zymogens. Those are zymogens. And so a classic example are the enzymes in your stomach, where there's an enzyme in your stomach which is, I forget which, there's like the pepsin and trypsin, things like that. I forget which one it is actually, and chemotrypsin. I think it's trypsin. I mean, there's a few of those are all, but I can't remember which is the one I'm talking about in particular, which is activated. It's inactive in its long form, but when the stomach acid increases, causes a segment of it to get chopped off. And when that segment gets chopped off, then it becomes active. And the idea being that it only wants to work when you've got food in your stomach so it doesn't digest itself. But were you saying that you remember which one it is? It's chemotrypsin? Okay, yeah, thank you. I can never remember which way around it is. But yeah, chemotrypsin is activated when the pH in your stomach drops in response to you eating food. So it is only active when you eat food. It also cleaves other digestive proteins to activate those ones as well. So you have several digestive proteins, and they all act as zymogens, and they all digest each other to switch each other on, which is kind of cool. Blood clotting works in the same way as well. It's often to amplify signals. Okay, and we've already talked about this, homotrophic versus heterotrophic. Inhibitors. Inhibitors can be broken down into two classes: reversible, irreversible. We're going to talk about reversible ones in detail. There's not very much to say about irreversible. There's like one slide on each that's just quite light. But we're going to talk about the four main types of reversible inhibitor in quite a lot of detail. Oh, this is the old slides. I've got to download the new slides. This is where it's going to vary now with you guys. So the new slides are here. And I believe inhibitors is the last thing we, yeah it is. It's the last thing we do, and then we will just do some examples of enzymes working until we run out of time, just for like a little bit of time. Screen 25, here we are. So, in the old slides, it is these ones. And you'll notice that this first one, it's got competitive in red and the rest of them are not. Whereas the new slides, it is this. So I just added 3 slides. But I think those 3 slides just give more explanation and make it more easier. Especially this one here. This slide here makes so much more sense when talking about enzyme inhibitors. And so on Blackboard, the PDF has been updated with the new version. And then if you've got the printout, those extra slides are printed off as a separate set of slides that you can have supplemental to go with it in case you're a printout person. Did all the printouts go? Is everyone go? Did anyone want a printout that didn't get one? Just one person? 2 people? 3 people? Okay, I'll print out more slides next time. Look, like I said, my undergrad class doesn't care about, so I print out like 20 slides. The class is like 45 to 50. I print out 20 slides and I only take like 1. When I come to this class and I print out like 10 and everyone's like, I want more. So it's just I guess it's a undergraduate thing, I don't know. Okay. So we're going to talk about the 4 different types of inhibitor, especially competitive inhibitors. Because once you understand competitive inhibitors, the rest just kind of slots into place. So, and this is why we talk about Lineweaver-Burke plot as well. It makes telling the difference between the 4 different inhibitors will give you a different type of Lineweaver-Burke, and so that's why the Lineweaver-Burke still has a lot of value. It either has value of just like having a quick look-see, or if you don't have the software, or if you just want to compare different inhibitors, it's really useful for that. So. There are the 4 different types of inhibitors. You can kind of think of them as being on a spectrum. You have on one side, you have competitive inhibitors. Competitive inhibitors only will bind to empty enzyme. Competitive inhibitors only binds the empty enzyme. And the reason for that is because they are competing for the active site. And if something else is in the active site, such as it is in the ES4, because something else is the active site, it's not competing. It only competes for the empty active site. And what that means is that if you've got 2 things competing for the active site, the inhibitor or the substrate, sometimes the inhibitor is bound, and so the substrate can't go in, and so the rate decreases. So that's how that works. On the other side, you have the uncompetitive. Uncompetitive inhibitors, they only bind to the enzyme in the ES. They only bound to it when it's already bound to some substrate. But what they do is that they stick it. They freeze it in the ES so it can't move on to the next step. So it inhibits the enzyme by stopping catalysis in the middle. It will only bind to the enzyme that's already bound to a substrate. That'll be an uncompetitive inhibitor. Everything in between is called a mixed type inhibitor, which I think the animation will come up to show that. Everything in between is a mixed type inhibitor. The last one remaining is a non-competitive inhibitor. A non-competitive inhibitor is a special type of mixed type. Where, so this is going to be in between. There's a spectrum. There's different inhibitors that will bind to everything in between. Will bind to both. Competitive will only bind to the empty. Uncompetitive will only bind to something that's already bound to a substrate, but hasn't moved on to the next step yet, and will freeze it at that stage. Mixed type combines to either, but it's a spectrum. It might prefer one over the other, or prefer the other over the one. It may have a preference. Non-competitive is just a special type that has zero preference. It will bind to either with zero preference. Whereas the mixed types, most of the mixed types will have a preference. Non-competitive is a special type of mixed type that has zero preference. And the effect that these inhibitors will have on enzymes is summarized in the table, and there's like 5 or 6 slides where I go into detail exactly how and why. But the short form is that competitive only binds to the empty enzyme, and it will increase the Km. If it has increased the Km, that means that it has lowered the affinity the enzyme has for its substrate. It has lowered affinity for substrate. But the Vmax, the maximal speed, is not affected. The reason why is just because it can be outcompeted. You may have an inhibitor that binds really, really well, but if you flood it with enough substrate, eventually you will hit Vmax. So it makes it so it's harder to reach Vmax. It takes longer to reach Vmax, but you will eventually get there because you can outcompete it. Let's say you've got 10 inhibitor and 10 substrate all competing for the active site. That may slow it down by a half. But if you've got 10 million substrate and just 2 inhibitor, well, the inhibitor's not going to have much of an impact. So it gets outcompeted, and you will still the enzyme will still be able to eventually work at its maximal speed. It will take longer to get there. Hence the increased Km, because it's the concentration required to hit half Vmax. Well, if you require more to hit Vmax, then you require more to hit half Vmax. So that's essentially it for competitive inhibitors. The uncompetitive inhibitor will decrease the Km, but it will also decrease the Km, which is kind of weird. Why would it decrease? Surely decreasing the Km is a good thing,
Yeah.
right? You would think. What it actually means, the reason why, is because, and I'm not sure whether or not I should save this for the actual slide that explains it, but maybe I'll just give you guys a little preview. The idea is that remember that E plus S to ES, that's in an equilibrium. Now remember, what does Le Chatelier's principle say? What is Le Chatelier's principle? I hope that the silence was because you all know the answer, but you didn't want to shout out all at once. Yeah. If you increase or decrease one of the sides of the equilibrium will work to get back to where it was. Now let's say that in the ES form, the uncompetitive only binds to the ES form, and when it binds to the ES form, it freezes it. It stops it from moving on to the next stage. When you freeze it and stop it from the next stage, that's kind of like actually removing it from the equation. It's kind of as if it wasn't there, because now it's just an inactive lump of nothing, or an inactive lump of something, I guess. Now, I've decreased what's on this side. So how's the equilibrium going to work to fix that? It's going to push it to the right. And so in that way, actually, you're pushing these together. You're making them so they want to react more. So the problem is that once they react, it doesn't move on to the next stage. So their affinity has increased. It's just that you can't do anything with that affinity because you trap it in the middle. You push it together, but then you trap it. And then because you trapped it, you remove it from the equation. Because you've removed it from the equation, you push it together more. But then you trap it and remove it from the equation.
So, is there like a new equilibrium established?
Yes, yeah. It'll keep trying to get to equilibrium, and then as long as there's an inhibitor around, it yeah, that's it. The non-competitive, notice that with the competitive and the, well, actually let's skip non-competitive, come to that in a moment. Let's go down actually to mixed type. Mixed type will reduce it's kind of got aspects of both. So we'll reduce the Vmax because it will do the same thing as the uncompetitive, and it will remove some from the equation. But whether or not it has the overall effect of decreasing or increasing Km will depend on which place on the spectrum it is. If it is more like a competitive, then it will decrease the Km a little bit. If it's more like an uncompetitive, it will increase the Km a little bit, depending on does it prefer to bind to empty, or does it prefer to bind to bound. That will affect whether or not it does the Km, what impact it has on the Km. And the non-competitive, it decreases the Vmax because it binds to it in the ES form and traps it there. But it has no impact on Km because it cancels out. Because it cancels out. Because it has an equal affinity for the enzyme in the empty or bounds to the substrate phase. Let's say that there's like 10 enzymes. Half of them will be inhibited competitively. Half of them be inhibited uncompetitively, and the overall effect will be cancelled out. You know, the Km will go up with some, the Km will go down with others, and then in the whole pool of the mixture, the effect gets cancelled out, so it ends up as a 0. And that's actually it. I'm now going to go through the slides that just explain again what I've just told you. But that's it for inhibitors. I'm also going to that's going to change the shape of the line we've about. If you understand what I just told you, that's all the question's going to be on. But you are going to be tested on this table. Not filling in this table, but rather I'm going to say, what's the difference between a competitive and a uncompetitive inhibitor? And expect you to just tell me this, this, and this, and that's it. Competitive does this, this, and this. Uncompetitive does this, this, and this. That's it. 5 points, boom, done. That's all you got to do. You see, it's not too bad. I'm sure that's what you were saying to the person next to you. Yeah. All right. So, like I say, I'll just go through the slides that are just explaining that. Oh yeah. So, this is weird, but I'm just saying, you know, like I was saying, it's weird that it brings the Km down. It's because the impact on the equilibrium. So, competitive type inhibitors, you have the enzyme. And it could bind with substrate, or it could bind with inhibitor. Competitive inhibitors affect the Km, but not the Vmax, because it's competing for the active site. But if you add more substrate, you can outcompete it. You can outweigh that the inhibitor is present. And so you will still reach Vmax, it'll just take longer to get there. And these 3 lines here, these 3 alphas, these are, alpha refers to the concentration of the inhibitor. And it's saying that the Km is increasing. And these 3 here, I prefer this diagram if they eventually came to the same Vmax. It doesn't, excuse me, so I think that Vmax is kind of rubbish. This diagram is kind of rubbish because it doesn't really show that. But these should eventually reach the same point. But the point is, you see that the rate at which they get to Vmax is much slower. So the rate of the V0 at the same concentration of substrate, the V0 is widely different. Let's say for 20, what's the units, micromolar, 20 micromolar of the enzyme, with 0 substrate, it's fast. With a 0 inhibitor, it's fast. With a bit of inhibitor, it's slower. With more inhibitor, it is slower still. So even at the same concentration of substrate, at 3 different concentrations of inhibitor, you've got it going slower. Because this is speed. This is faster, this is slower. So it's getting slower. The new Km is called the apparent Km, and it's given the variable AKM. So you have the Km for an enzyme, and you can say when it's inhibited, the apparent Km is alpha Km 5 instead of 2, something like that. 5 micromolar instead of 2 micromolar, something like that. But that's it. Remember that these 4 that I'm talking about, these are the reversible types of inhibitor. We'll talk about the irreversible, but there's not very much to them. They're quite simple and quick. So that's competitive inhibitors. Everyone good on competitive inhibitors? Competitive inhibitors are the easy ones. And this is, okay, and this is what the line we've about got. So, okay, so what are the, what's the thing that changes and the thing that doesn't change? Km changes, Vmax doesn't change. Now remember, if this is velocity, what is going to be the variable which is marked by the, it's shown there. But say if you think about it, like velocity is on the y-axis. So what is the other thing that's measured in the same units as velocity? Vmax, because they got the same units. So the intercept is Vmax. At the 3 different concentrations of inhibitor, concentration 1, 2, and 3, increase in concentration. What has happened to Vmax? Has not changed. But along the x-intercept, that is Km. And moving over onto, if you get here is a higher Km and here is a lower Km, you can see that the Km has changed. So if you're doing an experiment and you want to know what type of inhibitor you've got, let's say you've designed a brand new inhibitor and you want to find out what type of mechanism it has for inhibition. You can do an experiment where you get the enzyme without inhibitor, you do a few different concentrations and see what the rate is. A few different concentrations of substrate, see what the rate is. Do that same experiment but with 1 concentration of inhibitor, that same experiment but with a higher concentration of inhibitor, and if you want to be extra thorough, another concentration inhibitor. Then you just look to see where do the lines intersect. That will tell you what type of inhibitor you've got. If they intersect here, then you've got a competitive type inhibitor. So whereas if they intersect here, you've got a non-competitive type inhibitor. And the other types of inhibitors will come up in a moment for their different kind of, they have a signature look to them. But the lines intersect at the y-axis. Why? Because that is demonstrating Vmax has not changed, Km has changed. And also the slope has changed, right? Because the slope is Vmax over Km. So that's going to change and get steeper. Okay. Next is the uncompetitive. Wait a minute. Do we want to do uncompetitive or should we? Yeah, let's go to uncompetitive. This is the uncompetitive. This is the one where the substrate has to bind first, and then the inhibitor will bind. It will only bind. The inhibitor will not bind here. That'd be a competitive type. It will only bind here when it has already bound to some substrate. There should be a little S there, I think, to make that clearer, but just pretend that's there. So it binds at a site away from the substrate active site. It's not competing for the active site, so it's allosteric. Then allosteric. It only binds to the ES complex, and yes, it is still reversible. It will eventually leave. It's just that reaction can't proceed until it leaves. And it will decrease both the Km and the Vmax. And like I was saying, that's kind of weird. Wouldn't a lower Km be the opposite of inhibition? And like I said, the reason for it is because when the ES is inactive, you can consider it as not being part of the equation. You can remove it from the equation. And then because it's been removed from the equation, if this wants to be perfectly balanced, but you remove it, if it wants to be perfectly balanced, but you remove everything from one side, it's going to want to get back into balance again. And to get back into balance, some of this is going to turn into some of this. So it's going to move it across. It's going to make that first step proceed faster. So it's that first step is what is measured by Km. So it's going to bring it together faster. But it's just going to, remember, so Km is increased because of that. But if we go to the old thing, V equals K multiplied by the substrate. Well, K is not going to change, but if when the inhibitor has bound to ES, it decreases the level of ES, it's going to decrease the level of V, and so it's going to decrease the level of. It's kind of like when the Vmax is equal to K multiplied by E total, if the ES is considered inactive, it's kind of like the same as bringing E total down. If you bring that down, then you bring that down. So that's why the Vmax goes down. Because if this goes down, then this goes down, and if ES has bound to the inhibitor, then you bring that down. For the same reason as why you move this across. And then the Langweaver-Burke plot would look like that, which is kind of weird. And that is because both have changed. The Vmax has changed and the Km has changed. I'm going to show you some of these Langweaver-Burke plots in the exam. Now you could memorize them, or you can memorize the terms and then you can figure out what it is from the terms. Because you don't need to memorize that the three parallel lines make the uncompetitive. If you just know from that the uncompetitive reduces both, and you can see that this graph, both have been reduced, or they even just change. You don't even need to remember that it's reduced. If you just remember that it changes both of them, so I'm sorry, because it does, yes, it does reduce both. Then you can just see that both have changed. So it must be that. So that's what the Langweaver-Burke plot looks like for an uncompetitive. So already, if you run your experiment, you can tell the difference between a competitive and an uncompetitive.
So here the mixed inhibitor also changeable.
Yeah, that's true. That's true. But with the that's very fair. But the mixed it intercepts it intercepts, whereas the uncompetitive doesn't intercept. That's fair. That's fair. The mixed type inhibitor will bind to either. So if you have our empty enzyme, the inhibitor could bind to the empty enzyme, or the substrate could bind and then the inhibitor could bind. So the inhibitor can bind to E0 or ES. And so it's both. It binds to either. Now it may prefer, if it prefers the empty enzyme, then the Km will decrease. If it prefers the full enzyme bound to the substrate, the enzyme substrate complex, then Km is decreased. Km is increased. That's a typo. Wait, no. No, it's the other way around. There we go. There's the typo. And it will produce a Langweaver-Burke plot that looks like that. Well, it will just intercept off the axis. So yes, both of them have changed, but it will intercept off the axis for the mixed type. We've run out of time, so I'm going to move on a little bit. Finally, the last one is the non-competitive. It's a special type of inhibitor that will bind to either. So it will compete for the active site, or again, it will, once the enzyme is bound, it will bind allosterically somewhere else. So it will bind competitively and allosterically. But it has an equal affinity. And therefore, the effect on Km is cancelled out. This is because it decreases E exactly as much as it decreases ES. So it ends up cancelling out. And that one will look like this, because Km is not affected, but Vmax is affected. And often these are things in negative feedback loops. Comes up in biology as negative feedback loops. They switch off enzymes to say, we don't need any more. So I believe that's the types of reversible inhibitor. Just got 2 slides on irreversible that is very, very light. But are there any questions on the reversible inhibitors? It's a lot, but it's not awfully difficult. I feel like there's a lot of conceptual stuff, but it's not the most difficult stuff. No, you don't.
It is a lot.
Sorry.
It is a lot.
It is a lot. It's a lot of stuff, but it's not the hardest stuff. You just gotta. If you remember this, that always seems to happen. Like almost every day, there's always someone like jumping around upstairs. I don't know why. Psychology students, right? It's just probably doing some kind of experiment.
Yeah.
But this table really summarizes it and the reasons behind it. Especially if you remember, if you learn the reasons behind it, you don't need to memorize the table, because the table just tells its own story. Or even that comes in terms of like partial credit, things like that. If you just memorize it, but you get something wrong in memorizing it, then it's going to be harder to get partial credit. Whereas if you learn the concept and you write down the concept, but in the end you get something like mixed up the wrong way, I can tell from reading through to be like, oh, you got the answer, you just accidentally mixed up increase and decrease, but I can tell that you understood it, so I'll just give you the points anyway, that kind of thing. So I encourage people to learn the concepts, and I guess that's kind of why you're here, right? So you don't want to just memorize it, you want to learn things that will help you in your career. And you'll be surprised, you will be surprised when this stuff pops up again, like 10 years from now, and you'll be like, oh, I remember this. And now there's some money to look into this thing, and it involves using these inhibitors. And like, okay, now I've got to pull this out of my brain if I want that money. So you'd be surprised. Okay, irreversible inhibitors. There are two types of irreversible inhibitor. Type 1 is a type that will covalently bind. So rather than binding into a little pocket and making weak intermolecular forces like hydrogen bonds, static attraction, that kind of thing, some irreversible inhibitors will just come in and they will covalently bind. So you just get alkylation. This serine here will react with this diff-P molecule. And this diff-P molecule will chemically react and form a covalent bond, and that covalent bond can't be broken. And this serine, that serine may be important for the catalysis, and if you don't have that serine anymore, because it's been changed into something else, it can't do its thing. Maybe it's that reaction where something's getting phosphorylated, and that serine gets phosphorylated and then transfers that phosphorylation from ATP into glucose to make glucose 6-phosphate. Well, if that serine has been chemically reacted into something else, it can't do that anymore, and you can't reverse that. So that's the type of irreversible inhibitor. Those are often called suicide inhibitors because the molecule itself is gone now. With the reversible, they go in, they go out, they go in, they go out. With these ones, that molecule is gone, it gets used up. And then with transition state analogues, remember what I said, and I think transition state analogues is the last slide. It is. Remember what I said back here. The thing that fits into the enzyme the best is the transition state, because the substrate, if the substrate fit perfectly into the enzyme, there is no drive to turn it into the transition state. It's already super comfy. Why would it want to change into something else? More so, not only is it super comfy, so it doesn't want to change into something else, it is inhibited to do so because there isn't the space to. It would be, it's actually unfavorable, not just because it's already really happy. It's unfavorable because it would have to change into, there's nothing that favors it being in the transition state. Instead, rather, that enzymes are shaped so that they will bind the substrate adequately, not super effectively, but adequately. However, they are shaped so that they will fit the transition state perfectly, or particularly well. And then the trick is that then the substrate is going to bind and be like, you know what, this is kind of comfy. Do you know what would be more comfy? If I were to bend. And then it's a trick, because it bends, but then when it bends, it hits the transition state and it breaks. And then when it breaks, it's no longer the transition state, so it will leave. So it's kind of like a trick to trick the. I mean, it's not, because it doesn't have a brain. This is chemistry and physics. But it's like, you know, it's kind of like a trick where it's tricked into going into the transition state because it really nicely fits the transition state. But once it does, it turns into the product and then leaves. What if you had something that didn't turn into the product? If it didn't turn out into the product, then it will get trapped, because it only leaves because it turns into the product and is no longer the transition state. Transition state analogues are compounds where this is the, let me see, this is the, okay, so this is the transition, this is one of the transition states. This is transition state 1, this is transition state 2, and then this is product 1, and this is product 2. So actually this reaction here, does anyone know, does it say the name of the enzyme? It does not. Does anyone know what reaction this is, what common reaction this is, and what the name of the enzyme is? You're on the right track. Who was that? Edie? No. Yeah, you're on the right track. Not quite right. I know why you said PFK1. It's the one after. It's
What's
after
the
that,
question
right?
again, sir?
Sorry.
The question again.
Just by looking at this, can anyone tell me what process this is from, and what is the enzyme? The name of the enzyme. Sorry. It is from glycolysis. It's after PFK1 though.
Yeah.
It's aldolase. Aldolase, where the 6-membered sugar is being split into 2 3-membered sugar by retroaldol condensation. It turns as a G3P and D hat. Then phase 5 of glycolysis turns G hat into a second triose glyceraldehyde 3-phosphate. Anyway, transition state, product 1, product 2. Now, if this is the second transition state before it turns into product 2, this is what it looks like. The inhibitor is designed to look just like it, but not be it. Because these look similar, they will both bind really, really well into the enzyme. Because the enzyme is shaped to fit like this. The enzyme is shaped to fit this. So when this comes in and binds, it wants to become this. But if you design an inhibitor that looks just like that transition state, it will come in and bind because it wants to, because the enzyme wants to bind to the transition state. But because it is not the transition state, it's this amine alcohol here, as opposed to the aldehyde. Is this yes? Then it's not going to catalyze it, going to the next step and turn into product. So we'll just get trapped. It fits really, really well. I guess technically it's reversible. It's just really, 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 really difficult to get it to reverse. It just really wants to bind really well, and so it's really hard to get it out. So it's near irreversible because the enzyme is shaped to fit it so well. And that is that really. An example of that would be, for instance, so now I'm going to got like a couple of examples, which we don't really have time for. So I'm actually just going to, these examples are not things which are directly tested on, but these are things that may help you in the exam. So I will actually just very quickly just like read them out loud, so at least you have some context to understand what's going on if you want to study it yourself. An example of a transition state analog are inhibitors of HIV, so antiretroviral drugs. And the way that HIV works is that there is a HIV protease, which in order for the virus to duplicate, it needs to, at one point during its replication cycle, it needs to take a protein and break it into 2 via a protease. So this is a this is a peptide, and it's going to come in and hydrolyze this peptide into 2 separate peptides. And the HIV virus needs to do that in order to replicate. I'm actually not sure exactly where that is. I've not studied HIV, but at some point in its replication cycle, it needs to split a protein into 2 peptides. If it doesn't do that, it can't replicate. Now, what you can do is, what has been designed are these antiretroviral drugs, commonly sold antiretroviral drugs. Why can't I speak? And they are shaped. This kind of looks like a peptide, right? This looks like a protein peptide, but it is not. And so it looks like the transition state. It looks like this transition state here in the middle, and so it will bind. These will bind really, really well. The thing is that they are not peptides, and so they just are mimics of the transition state. So they will bind really well, but they will not turn into product, and so they get stuck in the HIV protease, and then the HIV protease can't do its thing, it gets inhibited, and the virus can't replicate. So that's like an example of those. This is just an example of some proteins that get regulated by different post-translational modifications. Things like phosphorylation, acetylation, different things that binds to enzymes, post-translational modifications to regulate their activity that a lot of the biologists are super comfortable with. An example will be, this protein that gets switched, on or off depending on when it's phosphorylated. So that's one way of regulating these things. This is just the zymogen. Like I said, it's inactive. And so this is the whole of the peptide from 1 to 45. But the chemotrypsin, Moses, you were right, the chemotrypsin gets cleaved, and the shorter version is the active version, like you were saying. It's exactly the same actually with blood coagulation. if you ever become a blood scientist, I despise blood science. You've got to learn all of this stuff. This is the, and if you do stuff to do with blood and the immune system, this is the clotting cascade. And there's like the intrinsic and things, intrinsic and the extrinsic. And this is the way that things like thrombin are different proteases that cleave other proteases, that cleave other proteases, that cleave other proteases, that cleave those factors, and those factors are what cause blood clotting. And it's a really complicated mess that I despise. But just for an example. This is just another example. I'm not even sure what I'm trying to say there. I think this is just an example of where the enzyme looks one way, but when the substrate binds, it will go induced fit. It will bind around it to increase the binding. This is just chymotrypsin. This is a multi-substrate enzyme where it's just saying, I think this is a question on the homework actually, and it says, the homework actually says, look at this slide on the, to get help with this. It says, what is the order of chymotrypsin working? Enzyme plus substrate to make enzyme products, product 1, product 2 leaves, product 1 leaves. That's just a multi-substrate, but rather than it being 1, 2, and then 1, 2 binding, and then 1, 2 leaving, one binds, splits into 2 products, and then those 2 separate products leave. So it's just E plus S makes ES, makes EP1, EP1P2, P2 leaves, P1 leaves, leaving behind E, and it just goes around again. Pre-steady state, steady state. Just examples. And different enzymes work at different, that's it. Different enzymes work at different pHs. Actually, that isn't pre-steady state, steady state. That's a whole different concept that I'm not going to talk about. We don't have the time. But that's it. Thanks very much, guys. Sorry I took an extra couple of minutes, but that is it. So we're going to do a class problem. I'm going to say, hey guys, here is an enzyme, here is a substrate, here is an inhibitor, just on paper. Tell me what it means. And I'll say, hey guys, here's an hour, tell me what it means. You can work together in groups to try to figure it out. And I'll give you hints and tips along the way too. But that's it. Thanks very much. Let me stop this.